Hello dear students. So now let's start with the last chapter of this semester, that is biochemical techniques. Under this chapter, or the syllabus of this chapter includes principle of biochemical techniques, then working and application of dialysis, paper chromatography, thin layer chromatography, column chromatography, gel filtration, ion exchange, affinity chromatography, electrophoresis, SDS page, and centrifugation. So all these biochemical techniques are included in this chapter. We are going to discuss about the principle, then procedure and applications of these biochemical techniques. Now, let's see what do you mean by biochemical techniques and what is the main principle of these techniques. Now, biochemical technique, it is defined as set of methods. Various methods are used, and these methods are qualitative and quantitative procedures. These methods includes qualitative and quantitative procedures. That means by using these techniques, we can find out the quality as well as quantity of the molecules present in the sample, and this enable scientists to analyze the substance found in living organisms. We know there are various biomolecules present in the living organism. Living organisms, their body is made up of various biomolecules like protein, carbohydrates, fats. So, and at the same time, various chemical reactions are also going on in the cell. To study all these biomolecules as well as chemical reactions and their products, these biochemical techniques help the scientist to analyze the products as well as the substances found in living organisms. As I told you, the chemical reactions also studied by using these biochemical technique. So, combiningly, how we can define biochemical techniques? That biochemical techniques are the set of methods, assays means which include qualitative and quantitative procedures that enable scientists to analyze the substances found in living organism and the chemical reactions underlying life process. Now the methods available for purification of biomolecules ranges from simple precipitation, centrifugation and gel electrophoresis to a very sophisticated standard methods like chromatographic and affinity technology, which are constantly undergoing development and improvement. All these biochemical methods or techniques, though they are very diverse, but they are interrelated methods. I can give you one simple example. Whenever we bring anything at home, we need to clean it, we need to purify it. But for all the things which we bring, we can't use the same cleaning process or purification method. For example, यदि आपने घर पे गेहूँ लाए तो गेहूँ का क्लीनिंग प्रोसेस अलग है, हमको उसको छानना पड़ता है, चुनना पड़ता है. बट वही यदि हमने वेजिटेबल्स लाए तो हम वेजिटेबल्स को वॉश कर सकते हैं वॉश करके ड्राई करके वी कैन क्लीन इट. यदि हम कोई पत्ता भाजी लाते हैं लाइक पालक मेथी, so we cannot directly wash it. What we do, first we clean it properly, remove the सॉइल, मिट्टी वगैरह निकालते हैं, फिर उनको अच्छे से पत्ता भाजी को तोड़ते हैं, बाद में उसको वॉश करके ड्राई करके रखते हैं. सो फॉर ईच एंड एवरी सब्सटेंस द टेक्निक्स आर क्वाइट डिफरेंट एंड इट डिपेंड्स अपॉन द सब्सटेंस which we want to clean. In the same way, for biomolecules also, the process of cleaning, it depends on mainly three factors. The first one is the size and shape of the biomolecule. There are two methods used to purify the biomolecules based on size and shape. One is dialysis and another is centrifugation or ultracentrifugation. Based on affinity, biomolecules has attraction. Some special affinity or attraction is present. So based on that affinity, there are two techniques. One is affinity chromatography and another is immunoaffinity chromatography. While the third factor which decide the process used for the purification of biomolecule is the charge present on it. Some biomolecules, they are neutral or some are having positive charge or negative charge, and based on that net charge, the purification techniques are electrophoresis. Under electrophoresis, we are going to discuss about SDS page and isoelectric focusing, while another method which is also based on the net charge of the molecule, that is chromatography. Here we are going to discuss about ion exchange chromatography and some adsorption chromatography. So these various biochemical techniques we are studying in this chapter. So again I am defining biochemical techniques. These are the set of methods which involve qualitative and quantitative procedures which enable scientists to analyze the substance found in living organism, as well as it helps to study the chemical reactions which are undergoing in the life processes. Now let's start with one by one all techniques. Now what is dialysis? Now dialysis, it is a process of separating molecules. As I already told you, dialysis and centrifugation, these two processes are based on the shape and size of the biomolecules. So dialysis is a process of separating molecule based on their shape, as based on their size specifically, and it control the movement of solute molecules by the difference in their rate of diffusion through a semi-permeable membrane from region of high concentration to low concentration. We know that smaller molecules can diffuse faster, while bigger molecules, they cannot pass through a pores of semi-permeable membrane, and so we can easily separate bigger molecules from the smaller molecules, because here the smaller molecules can move, these solute particles can move easily through the pores of semi-permeable membrane. Now let's see this diagram. Now here you can see that in this container, 1 minute. Now you can see in this container there are some smaller molecules as well as some bigger molecules. Purple colored molecules are bigger and green color molecules are smaller. And then these molecules are kept in one of the solvent, or it is a buffer solution of particular pH, which is also called as diacylate. And this bag in which solute particles are kept or molecules are kept, that bag is made up of semi-permeable membrane. Semi-permeable means which allows particular molecules only to pass through it. Now, when this bag is kept in the diacylate, smaller molecules, you can see green molecules, they move into the buffer solution or into the diacylate. And this movement is always from high concentration to low concentration. Now in bag, there is a high concentration of molecule, while in the solvent or diacylate, concentration is low. So this movement is from higher to lower. Now bigger molecules cannot pass through the pores, but smaller molecules can pass. And here in another diagram you can see that these smaller molecules, they pass through the semi-permeable membrane, and in this way they get separated from the bigger molecules. This process is continued till it reached to the equilibrium. Once it reached to the equilibrium, when the concentration in this container and the outsider solution, it becomes equal, the process gets stopped. If still some molecules, smaller molecules remain in the solution, then what need to be done? The process is repeated in another buffer solution. So in this way the process is repeated once, twice, thrice till all the smaller molecules get separated from the bigger molecules. Though this process, it is quite good to separate the protein molecules, bigger protein molecules from the smaller impurities, but it has some disadvantage. One of the biggest disadvantage is it takes longer time. Here we need to repeat the process continuously till we get the purest sample. And second thing, we cannot tell that by using dialysis we get 100% pure separation or pure protein sample. Here the separation is comparatively very poor, and that's why dialysis is not the good technique used in the laboratory. Now let's discuss, and again that's why it is not very popular technique. Another technique, it is a centrifugation. This method is also based on the shape, rather size of the biomolecule. Now in centrifugation process, first we'll see the definition of centrifugation process, that it is completely mechanical process, and here to separate the molecules, centrifugal force is used. In centrifugation, separation of molecule is done based on their size, shape, density and viscosity. The molecules whose density or whose size is bigger, they get settled down easily. Now see here, we'll see the process of centrifugation. In centrifugation, one device is used which is called as centrifuge. This is the device, it is called as centrifuge, and this device, it has the shaft, central shaft, and on this shaft it starts rotating. it rotates so fast that its rotations are 1000 rotation per minute. Means it can rotate on 1200 RPM. And at the same time, when there is a rotation, some centrifugal force is applied. Because of that centrifugal force, separation of molecules starts. Now you can see this is the container of the centrifuge, and in this container centrifugal tubes are kept. These tubes contain the sample which we need to purify or which we need to where we need to separate the molecules. Now, to understand the process easily, here I have taken one more diagram. Now you can see this is the centrifugal tube, and in this centrifugal tube the sample is taken. The sample contains some bigger molecules, some smaller molecules, and here we want to separate all these molecules from the liquid. So when this is the condition before the centrifugation. Once this tube is kept in the centrifuge, this device, and it is rotated, some centrifugal force is applied, and due to that centrifugal force, you can see during rotation centrifugal force is applied, and due to centrifugal force all the molecules, they get converted into pellets. They get converted into pellets means एक साथ जमा होना शुरू हो जाता है, और जब वो एक साथ जमा होना, कलेक्ट होना स्टार्ट होते, अल्टीमेटली दे बिकम्स हेवी, एंड व्हेन दे बिकम्स हेवी they get settled down, and in this way easily these molecules get separated from the liquid. So when they get separated, we get here we get pellets, and here we get the supernatant liquid. So in this way, by using this mechanical process, supernatant liquid or liquid get separated from solid. Specifically, this method is used in pathological lab. If we want to separate some molecules from the blood, RBCs, WBCs and other content from blood, it is a homogeneous mixture. But when it is kept under centrifugation, all the particles, blood particles, get separated from the plasma, and in this way we get separated molecules from the blood sample. Centrifugation has a number of applications, and it is frequently employed technique in the molecular biology or molecular biology laboratory. It is used to collect the cells, as I told you that in pathological lab for some detection there is very essential to collect the cells, so that can be done by using centrifugation, to precipitate the DNA molecules, to purify the virus particles, to study the viruses, their purification is very essential, and to distinguish the subtle difference in the conformation of molecules, which is not possible by some simple methods, but by using centrifugation, conformation of molecules, difference in the conformation of molecules can be identified. At the same time, centrifugation is a process which is simply used in some laboratories, but in genetics laboratory, ultracentrifugation is used, and which can be utilized to band plasmid DNA in cesium chloride gradient and differentiate various structures of replicating DNA in sucrose gradient. Actually, both these applications, I know you won't understand these. B group students, you have already studied these, but for A group student, I will request you that I will tell you that we will study all these in second term in genetic engineering. So till that time you just read it. Don't try to understand these two methods right now. So we'll stop here and the remaining part we'll see in next lecture. Thank you.